Good morning, everybody. Welcome to the first plenary session. I'd like to introduce my first co-chair here, Charlotte Gaydos, who's going to be the new IOSD president after this lunchtime. So that'll happen at the World General Assembly, and I'd like to encourage all of you to come. It's at 1 o'clock. It's in the program, exactly which room it is in. And those of you who are members of IOSD, please come along. The World General Assembly has to approve the final passage of the presidency to Charlotte. We anticipate that will happen without any problems. And Charlotte will be introducing the second and third plenary speaker to you. I'm David Lewis, the current president of IOSD, and I have a few announcements before we start. First of all, our third speaker has elected to speak in Portuguese this morning. It's quite an emotional thing for her to do that. She really wants to. We've been trying to sort out about the headsets, which is why we're a few minutes late starting. That process of trying to sort it out is still ongoing. We will let you know by the end of the second plenary whether or not headsets can be made available. If not, I know the slides are in English, and I hope that you will bear with us and be able to follow the slides in English while our speaker delivers her lecture. The next thing I'd like to make an announcement about concerns the program, the scientific program. So we've become aware that several talks and several speakers' names are missing from the hard copy of the program that you have in your bags. The online version at the conference website is the correct and updated version. And so we really encourage all of you, where possible, to use the online version to make sure you don't miss out on any of the talks. Okay, so before we get started for the IOSD prestigious lecture, we're going to have a little bit of a discussion in Portuguese, just as an elementary lesson today. And I'm going to discuss with you about the Carnival Ball, which is happening tomorrow at 7 o'clock. We leave here in the buses. The last bus is 7:15 in Brazil, is mais ou menos. So sometimes that means a little bit later, but don't risk it. Please be there by 7:15 latest. Now, the Carnival Ball, you have to buy a ticket. It costs $80 US, $80 dos Estados Unidos. So please make sure that you get your ticket. You get them from the registration desk, okay? Now, what else to tell you? It's going to be quite local. It's at Casa Shopping at the Fogo de Chão restaurant. So if you've not had a Brazilian churrascaria type meal before, this is your great opportunity. Lots of salad and lots and lots and lots of meat. But the vegetarians, the salad is quite an amazing bar, so you should have some options there. So that's very near. The buses will bring you back as well, so it will be safe. Now, the reason I put these lessons up here, because this is the theme. So you can come sparkly or brilhante if you are going to learn it in Portuguese today. Fabulous, or as Basil, my friend from Sydney, would say, fabuloso, which is a very lovely word. Creative or creativo, and gala, which is the easiest one for everybody to remember. Gala is gala. And just in case you don't know what sort of things you can wear, I thought I would just show you a few pictures. It can be very simple. Very simple, just a hat or something sparkly on your head, or get a little carnival mask. And there are some shops up there in the Casa Shopping Centre. And also Monica, who works for the conference, has got a little bag she's been running around with some various things with feathers on. Please, you're entitled to get that. If you want just to colour yourself, that is creativo, okay? Then, Jeannie, this is eleganci, or this is the sort of beautiful styles that you can wear with the mask. If you really want to start pushing out for fabuloso, this is fabuloso. And this one is a friend of mine when we were in Bahia, and he went as melazinha, which is basically his little melon. And if you're like me and you're careca or you're bald, then you can use beautiful wigs to cover up the gaps. But most of all, you must come and have some fun. Now, we have another special announcement today, and this is why I wanted to get you into the mood of your Portuguese, because we have a special announcement. And nós vamos cantar agora. We are going to sing now a little bit, because today is Ginny Marazzo's birthday. So we're going to do it after 3. We're going to sing it, and instead of saying happy birthday, we sing parabéns pra você. And when we get to the 3rd line, it's very important, it's Ginny, okay? So we're going to sing this in Portuguese together. After 3, 1, 2, 3. Parabéns pra você, parabéns pra você, parabéns pra Ginny, parabéns pra você. And so it's a great delight for me to invite to the platform Ginny Marazzo. Ginny is going to deliver the 2nd UST prestigious lecture. Those of you at the opening ceremony heard the glittering account, which I'm just going to run quickly through. Dr. Ginny Marazzo is Professor of Medicine and Director of the Division of Infectious Diseases at the University of Alabama in Birmingham. She is internationally recognized for her research and education efforts in the field of STIs, especially for those areas of the field which affect women's health. She's a fellow of the American College of Physicians and the Infectious Disease Society of America. She's also the chair of the American Board of Internal Medicine Council and also chairs that council's Infectious Disease Specialty Board. She's board certified in infectious diseases, and she loves to conduct her research on the microbiome. So I'm going to leave it there and save any more embarrassment for Ginny. Please come up and give us the lecture. Thank you.
What a wonderful way to start my birthday. Thank you. I was trying to do it on the down low, but clearly that was not meant to be. Charlotte Gaydos, thank you very much for letting everybody know it is a good birthday. It's a great place to be here. So I could not be more honored and more humbled to be part of this all-girl plenary session, I want to point out. I have two fantastic speakers succeeding me, and I'm going to update you on the vaginal microbiome somewhat selectively. Actually, it was in London when the organizers kindly invited me to do a plenary, and at that plenary I talked about the new molecular methods for defining vaginal microbiome. That was very new at that time, and we have come a really long way. So let me start by trying to take you through some of this, and I'll explain the selection of the content in a moment. These are my disclosures. So what's new and what am I not going to talk about? Okay, so what I'm not really going to talk about very much is treatment of BV. And for that, I would refer you to the presentation on the new Lancet Commission on STDs, which is going to be presented today from 12:30 to 1. Kat Bradshaw is actually going to present the part on the bacterial vaginosis section that Jane Schwebke and she and I put together, and that really is a call for new approaches to treatment and managing the vaginal environment. So I encourage you to take a look at that. I'll also point out that there are a couple of things, there are a couple of trials going on. There's a trial that just finished looking at a novel biofilm disruptor that I ran along with Jane Schwebke and Julie Dombrowski in Seattle. Results should be coming in the next 6 months or so, so that's exciting. I know we're not supposed to use the lactobacillus in yogurt to treat vaginal problems, but I couldn't resist this lovely image. And it's actually from a great article in BBC Focus, Science Focus magazine, that interviewed a number of people in this room and talks about the promise of probiotic treatment for BV, which, as you may know, is being assessed in a trial led by Craig Cohen right now using the lactin V, Lactobacillus crispatus. So these are things that we can anticipate in the coming months to years, but they're not ready for prime time yet. So take a look at the information of those on clinicaltrials.gov. And since the last time we were together, we have had the first approval of a nucleic acid-based amplification generation BV-associated bacteria diagnostic test, namely the BD-MAX vaginal panel, that has a very sophisticated algorithm for detecting quantities of Lactobacillus crispatus as well as BV-associated bacteria. So very exciting, and a lot of presentations on that that you should be hearing about today. So stay tuned. But I'm not really going to talk about those things because, unfortunately, and that's what the Lancet piece really emphasizes, we don't have a lot that's new in treatment of BV, and that is what is so challenging and so challenging, basically, about this problem. So what am I going to talk about? I'm going to sort of revisit what a healthy vaginal microbiome is, because I think we do have more data to understand that. And I'm going to focus on the consequences of not having it, particularly with regard to the increased risk of HIV acquisition. I loved Kevin DeCock's plenary last night. I think his comments about convergence and divergence of STI and HIV set the stage perfectly for this, because I've always felt like, you know, HIV was really driving the wave, riding the wave. STIs were towed behind this little life preserver. Now, between the emergence of STIs in the era of PrEP, plus some of the data I'm going to show you on vaginal microbiome and HIV acquisition, I think we really need to be up there on the wave with HIV, and that's a great metaphor considering where we're at. So I'm mostly going to talk about that, and some of this is data that either hasn't been published yet or was literally just published in the last month, and I think if you haven't been going to some of the HIV meetings, you may not have heard about it. So I'll touch on that. And then I'll just close by thinking about some ways that we might start innovating in our approach to maintaining the normal vaginal microbiome and defining pathogenesis, which we have really started to do. So just a reminder, I know not everybody thinks about the vaginal microbiome 24 hours a day like I do. In fact, I can't think of anything I'd rather be doing on my birthday than giving a plenary on the vaginal microbiome. Remember that there are many benefits to the optimal vaginal environment, and I'll talk about that in a moment. The main things are that you have protections from pathogens and you don't have vaginal symptoms that are referent to bacterial vaginosis. So you're less likely to get BV, HIV, chlamydia, gonorrhea, trichomonas, and there are really great prospective data, even from the cultivation era, to show that. You're also, you're less likely to transmit HIV, that should be transmitted, and if you have HIV, you probably have lower levels of genital HIV. The main reason that people originally got started looking at BV was because of the pregnancy-related outcomes, and that before HIV, those were really the big things that people thought about. And we know that there are now optimal birth outcomes associated with a lactobacillus dominant microflora, and that would be crispatus or Jensenii, that are not just short-term, meaning fewer pregnancy-associated infections, normal birth weight, normal timing of delivery, so fewer preterm deliveries, but also long-term. And this is data that has really blossomed. The field has really blossomed since the last time we talked about this. For example, we now know that infants born through a healthy vaginal canal, particularly as it relates to C-section born infants, are more likely to have lower rates of autoimmune disease. I think the data are better for asthma than anything else. And we also now have really interesting data to say that as soon as a baby is delivered through a healthy vaginal microbiome, there's immediate colonization of the baby's oral cavity as well as the gastrointestinal tract and the genital tract with the mom's healthy vaginal lactobacilli. I can think of no more intriguing example of this than Gloria Dominguez-Bella's lovely, small, but really intriguing study that was published in Nature Medicine just last year. And this follows on her study in PNAS a couple of years ago that showed that delivery mode shapes the initial microbiome of the infant. So if you're born by C-section, what are you initially colonized with? In your gut as well as your skin? You're colonized with skin flora, largely staph, Corynebacterium, diphtheria, that sort of thing. If you come through the vaginal canal, you're colonized with Lactobacillus species, or the species of BV if you've got a microbiome disruption. So that was the first sort of proof that really kind of established, I think, definitively that that was the way that you are initially colonized, and really important for immune modeling. That's probably why these long-term outcomes are so critical. What she did then was this really intriguing study where she took 18 infants and their mothers There were 7 born vaginally and 11 delivered by scheduled C-section. And what she did with 4 of the C-section baby-infant sort of mother pairs was to colonize the babies born by C-section with the vaginal secretions of the mom, assuming the mom had healthy vaginal secretions, and showed that immediately those babies were colonized throughout their body with the helpful or healthy vaginal lactobacilli relative to the C-section babies. The prevalence of C-section in the United States is about 32%. I just saw the updated statistics from CDC or whatever. So you can imagine that this is potentially a huge impact on the baby's immune system and as they age. And you know we're having a lot more problems with asthma and allergic-mediated issues. So fascinating data, just a reminder when we come back at the end of the talk to thinking about innovative changes in the vaginal microbiome. Now for those of you, again, who need a little refresher, remember the spectrum of the vaginal microbiome that I've already alluded to was defined very early on, largely with Gram-stain and cultivation, and on the left there you can see the beautiful lactobacillus predominant, which would be a Nugent score of 0, and on the right that anaerobe predominance with clue cells and a mix of anaerobes. Since the time that we've met in London and elsewhere, use of molecular analyses to define this environment has really pretty much become the standard. And in general, what I think is intriguing is that they pretty much continue to complement the traditional methods. If you look at what we found out from the molecular analysis, and Sharon Hillier would be delighted to hear me say this, there was an initial surge, I think, of excitement. Maybe we would find the pathogen associated with BV. I don't think we've really done that. We've certainly clarified and detailed the community, but I don't think it's been quite the holy grail that we thought it was originally. I'm also going to talk a little bit about newer methodologies, mostly proteomics, to define some aspects of the microbiome. The bottom line is that all of these methods are defining a spectrum from what I will call optimal to less optimal. I started even thinking about not using the term BV because, as I'll talk to you about in a second, I think that BV is a simplistic and reductive term, and I think we're talking about different syndromes or different actually microbial communities that probably impact different local inflammatory responses, and I'll come back to that in a second. But the bottom line is that in the optimal vaginal microbiome, and I define optimal as healthy birth outcomes, lower rates of infection, whether you're talking about it in the reproductive tract or acquisition of infections, that's an optimal lactobacillus, Jensenii or crispatus, pH acidic environment. That's really what it is. This also gets you away from talking about, well, if 70% of women in Africa have BV, isn't that normal? That may be the norm, but it's not optimal when you talk about acquisition or risks conferred by that environment. So it gets you away from that sort of more, I think, culturally very important thing to think about. What's normal, what's the norm, and is that the best thing for the person involved? Okay. This is just a paper I'll use to introduce this concept of diversity of the vaginal microbiome, and beautiful work by Sujatha Srinivasan in David Frederick's lab. Sujatha's here. Always like to present this, and this is very similar to what Jacques Ravel and others have shown. So you could really choose papers with any of these groups and you get a very similar thing. And what you're looking at here is 16S ribosomal DNA, PCR, and PIRO sequencing of a couple hundred women. Each of these lines of colors is an individual woman, and you can see the AMSL score is under the letter A, the Nugent score is under the letter N. If it's red, it's abnormal. If it's green, it's normal. And essentially what you're seeing is that there is sort of a bimodal split. Everybody down below who is supposed to be normal clinically by AMSL and Nugent seems to be predominated by those two colors, orange and red. Those are Lactobacillus crispatus and our very confusing friend Lactobacillus inners, which I'd like to come back to because we really don't know what Lactobacillus inners is doing. It doesn't make lactic acid, it doesn't make the vagina acidic, and yet there's a lot of it around, and in fact you can be completely quote-unquote normal, at least by our crude measures of AMSL and Nugent. What I think is even more interesting, if you look at the top, the women who have BV defined by those criteria, you've got some clearly distinct groups. You can see the women on the top are dominated largely by that green bug, which is Snethia leptotrichia. You may remember that that has been associated with urethritis in males. So you may wonder, are those a group of women who have acquired sexually associated BV because their male partners are continually exposing them to Snethia and leptotrichia? Absolutely. And then you can look down and see the pink-dominated people. Those are Gardnerella vaginalis and et cetera. You can sort of keep going and looking that there are really diverse heterogeneous communities. So I think, again, this is only 200 women. It's a selected sample from the Seattle STD Clinic. If you start to do this on a larger scale, could you really start looking, and especially when you start looking over time, could you really start looking at some of these communities as fingerprints or different predictors of things? And again, remember, women who don't have BV are dominated by Lactobacillus iners or crispatus. The other thing that's in this paper, which I think is also very important and draws the link between just describing the presence of those bugs and the endpoints of symptoms, or in this case, the EMSL criteria, is shown here. This simplicity of this slide belies a very sophisticated analysis where Sujatha and her team of computational biologists and statisticians went in to look at what bacterial taxa were associated with the individual EMSL criteria. And it turns out that the really big players here that affected all components of the EMSL criteria, which are abnormal pH, clue cells, an amine odor, and vaginal discharge, are Gardnerella vaginalis, Leptotrichia amnionii, again, getting back to that Snethia leptotrichia group, Egerthella, and Adipobium vaginae. So, and those are going to be critical, so keep those in mind. And I think this is the kind of thing we need to start doing on a more sophisticated basis. So my conclusion from this body of work, not just Sujatha's, but everybody's that's been working in this area, is that all BV is consistent in having elevated pH and loss of non-inners lactobacilli, meaning Crispatus and Jensenii, which likely accounts for the consistency of adverse reproductive and sexual health outcomes. So to me, that's a common pathway, right? The environment is disrupted and it's got those things. Otherwise, I think that there are probably discrete types of BV, and this is totally a hypothesis on my part, although I'm not the only person who has thought about this, that may be defined by community composition of individual species with direct impact on local immune environment. And I would call that an individual pathway, and that may explain, not just because of the microbes involved, but also because of the host response, why not everybody with BV has the sequelae that we think about. So I think that's worth thinking about. So I want to shift a little bit, and I want to come back to Kevin's talk last night. This is a group that is the World STI&HIV Congress, so I want to shift to talk a lot about much of the talk about this relationship with HIV, because I think this is a perfect lens to start looking at this diversity question and the host inflammatory response. So just a reminder, this is an old meta-analysis, but I still really like it. I think it was done well, and it's often quoted from Adeshili et al. In 2006, I think now. And I think it's been borne out by many subsequent smaller analyses. And the consensus is that women, again with BV, just let's use that term with the caveats I mentioned, have about a 60% increased risk of acquiring HIV infection through vaginal sex. And that's if you control for everything else. STIs, types of and frequency of sexual behavior, not controlling for partner's viral load. What's missing in all these analyses is the male partner, which I think is a huge weakness, and we have not really come to terms with that. But I think just across the board, you can use this 50 to 60% increase as a pretty robust example. Many of you may not recall that Craig Cohen from the Partners in Prevention study also published a very nice paper in PLOS Medicine a few years ago showing that women with BV were also more likely to transmit HIV to their male partner. And this was great because there actually was a biological gradient with increasing abnormality of the vaginal microbiome. So as your Nugent score got more and more abnormal, the risk of a woman with HIV transmitting HIV to her partner actually went up to the point of, as you can see, about 7-fold. they tried very hard to make this association go away. It's a beautiful epidemiologic analysis for those of you who want to try to understand methodologically how complex this is. They controlled for everything. I'm hiding it there by my summary box, but they really tried to control for everything, and they did not make it go away. What's the main hypothesis that would have explained this? That the women with BV had higher rates of genital shedding or higher genital HIV load, and that explained it. Even when they controlled for that, they couldn't make this go away. So it's still something else that's going on, okay? I also like to refer back to this really nice analysis that I think hasn't gotten enough attention by Lynette Masese with Scott McClellan and their group. And what they did was to revisit data from the Mombasa cohort that University of Washington research staff have been following for over 20 years now, actually closer to 30, I believe. And what Lynette did was to look at 20 years of data and look at HIV acquisition in this cohort of women and attribute the increased risk of HIV acquisition to the conditions that these women experienced over time. Incredibly unique opportunity to look at a database with the same women over 2 decades where vaginal infections and STDs were monitored throughout. What did she find was, regardless of the time period, you can see they're broken out by 4-year periods there. Of course, genital herpes, right? Acquisition or prevalence of genital herpes had by far the highest population attributable risk percent. So, of course, we can't forget that. But the next most important thing was bacterial vaginosis. And again, that accounted for about 20% over time. Absolutely no difference over that time period. So when you think about the prevalence of BV in these women and you think about the incidence of HIV acquisition, you're talking about an incredibly important attributable risk percent. So worth taking a look at that. How does this happen? Well, this slide I put together a while ago, and I updated pretty frequently, although there's now so much data that I can't really put everything on it. I'll just try to sort of point out that there are some general principles that people have postulated mediate this increased risk of HIV. Clearly, you lose your hydrogen peroxide production by your good lactobacilli. Hydrogen peroxide is directly virucidal. Alkaline pH actually can directly activate CD4 cells. Also, you can get activation of Langerhans and CD4 cells directly by these bacteria. And then the cytokine story. Now, the cytokine story could be related to all of this, but there are really interesting accruing data. We've always known that cytokines are disrupted with BV, but it does appear that some of these really promote local HIV replication and increased shedding. And there are a number of studies there that you can take a look at. Rupert Call's group very early on showed that successful BV treatment actually reduces some of these disrupted cytokine and decreases activated CD4 T cells at the endocervix, which is really very interesting. It's been tough to replicate that, so we'll talk about that in a second. Let me see if I can maybe go back. I had some trouble last night. Okay, so that is just some idea about what's going on. I would say where we're at right now is that we finally have some new data to help us understand this mechanistic link. It begins with inflammation, and I put that in quotes because everybody has a different definition of what inflammation is. And it ends in some confusion, which is how I'm going to probably end this. But it is a start, and I think it's a really exciting thing for us to start thinking about. So this is an older paper, too, by Andrea Thurman at Conrad with a number of colleagues. But it's a nice paper if you haven't really started to delve into this concept of how BV modifies the local immunology. One of the key things I think is important is that IL-8 is increased. There is production of sialidase, which actually destroys IgA, and there is IgA in the vagina, and in fact, there is IgA specific to Gardnerella vaginalis. Sabina Cocci a long time ago showed this. So again, all of these threads coming together. So many ways that you could conceivably postulate that this environment would be very conducive to BV. So we've known all of this for a long time, but again, putting this together with real-time HIV acquisition studies has not really happened. What I think is new since those papers are published is that we've started to get into an era where we're using functional assays, and I'll describe that in a moment, as well as omics. And by omics, I mean metabolomics, proteomics, that sort of thing, to really start looking at this a little bit more carefully. Betsy Harold's group at Einstein has done some very nice work using an assay that looks at activity of vaginal secretions, vaginal fluid, against E. coli. It's not really clear what confers this E. coli effect, but it seems to associate very strongly with healthy features of the healthy vaginal microbiome. Lots of work coming out on that. The other thing that that group has shown is that that inhibitory activity is associated, when you use proteomics, with proteins produced by L. crispatus and L. jensenii. So I think it's very likely that these bacteria are producing something that is actually promoting this healthy and protective effect. Later data from the Rupert Call paper presented at the HIV Research for Prevention meeting in 2016 again emphasized that when you've got these cytokines associated with BV, you not only increase your endocervical CD4 T cells, which increases the target for HIV, but you also, when you use these new proteomic approaches, get evidence of epithelial disruption. So you put this all together, you've lost your hydrogen peroxide, you are not producing these proteins that seem to be associated with protection or at least functional anti-E. coli activity, which has been associated with vaginal health, and you may be disrupting the vaginal epithelium at a very, very microscopic level that we can't really see. It's not like a vaginal ulcer. I think this is starting to put together the picture that we might be trying to tell ourselves about why this is happening. And I would refer you to a nice review paper by Rupert that talks about this, that again is a little bit of 2015, but really goes into some of these issues in detail. Now, people got really interested in this last summer, a year ago, because at the Durban IAS meeting, the CAPRISA group, as well as Scott McClellan's group from University of Washington, presented some fascinating data. And this is where I would say people started finally to remember that HIV is acquired in the vagina. Isn't that like amazing? The virologists were just like, wow, really? I can tell you a funny story about that later. So what did the CAPRISA group present that was so interesting? And this data has finally just been published. Nikki Klatt published it in Science just June 2nd, I believe, and I'm going to show you some slides for that. This group had shown previously, that's the Melisana JID paper from 2012, that the women in the CAPRISA study, and for those of you who don't remember, CAPRISA was a study of pericoital tenofovir gel versus placebo gel for prevention of HIV acquisition, and it showed a reduction of HIV infection in the gel users by about 50%, and in the highly adherent users about 68%. So it actually worked in that study, much to many people's surprise. That group had generated what they called was an inflammatory profile, and the top picture, which you're not supposed to see, but just a reminder for me to say it, is that they had measured a large panel of cytokines and defined a group of cytokines that they were confident defined an inflammatory environment, and that inflammatory environment by itself was associated with HIV. And that's what this graph shows you, or this table shows you. The women who had genital inflammation, again is defined by, I would say, a rigorously defined cytokine score, had an odds ratio of 3. Is that surprising? Not really. We've known for a long time that having an STI, right, particularly TRIC, or an inflammatory, even Candida, actually vaginal, is a risk factor for acquisition of HIV. But interesting to see this with a very sophisticated cytokine analysis in a group of young women in sub-Saharan Africa. So where did they go with that from there? What they then did, which is new, is with the help of Adam Bergener's group in Manitoba, using proteomics, not using 16S, ribosomal DNA analysis, sequencing, or anything like that, and I'm going to come back to that issue. They then went in to look at what the microbiome was that was associated with this inflammatory score. And what you're looking at here is horizontally a spectrum of community types. So remember the paper that I showed you before with Sujatha's nice graphic where all the community type, the diverse communities associated with BV were at the top, the more homogeneous communities were at the bottom. This is just a horizontal version of that. The women on the right with all the colors are getting into increasingly diverse community states, okay? And you can see here that the community cluster or community state type 4 was linked most strongly with this genital inflammation and HIV. And that was, again, using this proteomic definition of the microbiome, okay? What was in that cluster when they looked more carefully, again, not by specific PCR, but by this proteomic approach, was Prevotella bivia. Prevotella is one of those big bad anaerobes of BV. If you go back and look at the old prospective data that Sharon Hillier, Dave Eschenbach, others did of adverse outcomes in pregnancy, Prevotella is one of those black anaerobic gram-negative rods that was associated with worse outcomes of pregnancy. But fascinating, here it is again, 25, 30, 35 years later, coming up in this proteomic analysis. And what they showed was that P. bivia was strongly associated with genital inflammation and HIV acquisition. So if you had P. bivia in your vagina, nothing about quantity, nothing about PCR specificity, all caveats, and very small numbers, so keep that in mind, please. 19 times more likely to have genital inflammation and 13 times more likely to acquire HIV. Now, this is a lesson in interpreting these data with caution. What was the initial reaction to this? Oh, we need to find antibiotics that will kill Prevotella bivia and start infusing them into the vagina continuously. That is not the appropriate reaction, right? You really have to take a step back and try to think about what is going on here. Is this a marker for something? Do we need to look at relative quantities? Is there a community associated with it? Are there behaviors that are associated with this that are actually the true mediator of this phenomena? No idea. So lots of, frankly, scientifically, I think, irresponsible knee-jerk reactions to these data were evident. So at the same meeting, that got a lot of press, a lot of reaction. At the same meeting, Scott McClellan and colleagues, as I mentioned, of the prior Mombasa data, showed a very similar analysis, which was not a talk, was a poster. And what they did was to gather data from a large cohort of women from several settings in Africa. Many of them were pregnant. I think about a fifth of them may have been in pregnancy studies. And they did a very careful statistical case control, nested case control analysis that Sujatha actually led the laboratory work on to look at a very similar question. When you look at the vaginal microbiota in these women, prior to HIV acquisition, can you look at cases who acquired HIV versus controls who did not and identify some signature disruption in those cases that could explain why these women got HIV? So this is what it looked like, and they characterized the microbiota by deep sequencing and qPCR. So very different approach. What did they find? Well, not surprisingly, the cases versus the controls had more diversity, right, in their vaginal microbiome. Again, that's really, to me, not very different than saying BV is a risk factor for HIV. That's really what we've been saying all along. And I should mention this has not been published yet. It just was presented. So this is just the diversity index in the cases on the right versus the controls on the left. And you can see the individual bacteria there that were looked at in this analysis. When they went in there with specific quantitative PCRs, so now getting much more refined than the previous analyses, to look at whether you could find a predictor by quantity of those vaginal bacteria, you can see that they did find some interesting trends. Egerthella, remember, which was in that confluence of the AMSL criteria, came up actually as a risk with a significant p-value. A couple of others there, including our friend Leptotrichia snethia, not surprisingly maybe, because we know that's associated with urethritis, and there's going to be a nice presentation on this, I believe, today. And then Mycoplasma hominis, not just barely significant. So This did not show Prevotella. They looked very specifically for that. So what does that mean? Why is it different? What can we say? You have one group saying that we need to go in and target this bacteria and maybe even develop a vaccine for it. And then another group that's saying we really don't know what's going on here. Well, I think there are a couple of different reasons. And again, this is why I would really urge caution, and when I come back to this at the end, talk about where we need to go from here. Very different populations of women. There are lots of unmeasured variables. And as I said, we have no data on exposure to male partners' microbiome. And I think until you put that into this equation, talking about sort of drastic modifications to the vaginal microbiome is not particularly thoughtful. But let's come back to that. As I mentioned, they use different techniques to define the microbiome, and this is part of the evolving scientific landscape, right? The traditional, now traditional, it's traditional since not very long ago, method is really the deep sequencing, and that's what Scott and his group did to direct and define quantitative PCRs to look at these bacteria. The Caprisa group used this proteomic approach. What does that mean? They basically search for bacterial peptides, so proteomics peptides, that were then associated with a database derived from earlier 16S ribosomal RNA work, okay? So it's derivative. It's not wrong. It's not less robust. It just is a different approach, and you got to understand the limitations of it. And again, there wasn't specific quantitative PCR. They estimated bacterial abundance by summing these protein spectral counts, which is very creative, but I think needs more validation before we really sort of go to say that this is what's going on. Now, on the heels of this fascinating paper that I think you should know about if you haven't seen this, by Doug Kwan's group, this is Christina Gossman's paper that came out just earlier, I want to say, let's see, what are we, 2017? It's earlier this year, February of 2017. And this is an interesting paper because it took advantage of something called the FRESH cohort, also in South Africa. But what's interesting about the FRESH cohort is that it is very young women, teenagers, essentially. I think maybe up to 21. And I'm going to tell you what they did here. They looked at 236 young women, and they defined 4 vaginal community types at baseline using more traditional, quote-unquote, sequencing methods, as I mentioned. And again, this is not surprising, right? If you look at these community state types, this is pretty much what I showed you before. You've got type 1, which is dominated by Lactobacillus crispatus in orange, type 2 in green, that pesky Lactobacillus inners, and then increasingly diverse, some would say disrupted, vaginal microbiota to the point where you go all the way to this community state type 4, okay? So keep that in mind. I do want to point out in the community state type 4, that sort of mauve color that's dominating in the middle range of the drawing, that is Prevotella species. So it is there, and it's going to come back to haunt us, okay? What they did then was to assess whether these community baseline types predicted acquisition of HIV in just 31 women, okay? Only 31 women who are cases. So again, caveat. They sequenced the V4 region, and they also measured immune mediators, and then novel, they did an associated animal model assessment in the mouse, and that's what I want to show you. There's a couple of really fascinating findings from this. They looked at chlamydia, they stratified by chlamydia. They didn't really, chlamydia was associated with an increased risk of HIV acquisition, actually a 4-fold risk. So for those of us who are still interested in chlamydia, that's a pretty important finding. But what I want to show you is the small graphic in B. And this breaks down the HIV acquisition by those community state types. There are a couple of stunning things to me in this very small table. Again, small numbers. No one who had community state type acquired HIV in this study. The incidence was 10%, I think, 8 to 10%. I can't remember what it was, but it was very, very high. Well, it's 31 women out of 200, so that's what it was. None of those women acquired HIV. 112, really, really impressive. Of the group of community type 2, the inners group, 9. So this, I think, again, tells you that inners is not really helping here. What's it doing, and why is it associated with a normal clinical environment? We don't really know. Look at the acquisitions in types 3 and 4, particularly type 4. Really, almost all of the acquisitions occurred in 3 and 4, but nobody in 1. So I think that's really, really very, very telling. And the other things that they showed in assessing the immune environment in those women was that when you had this community type 4, and again, getting more sophisticated from the old sort of just straightforward cytokine analyses, was that they had more HIV target cells in tissue. They had elevated levels of MIP1 alpha, MIP1 beta. These are CCR5 attractants. So you're, again, attracting the target cells. Not surprisingly, our old friend IL-8, IL-1 alpha, TNF alpha, IL-10, and also IL-17, which is a newer sort of dimension. So really nice, I would say, work. The last thing that they did, which was kind of cool, and that's just the first 3 points are just some summaries, is that they took Prevotella. This was sort of separate, but they were going down that Prevotella road, again, because of what Caprisa had shown, and again, because of the Prevotella dominance in community type 4, and they gave it to mice, and they put it into the vagina of mice. Mice are not necessarily a great model for the human vagina, but they will do. That's one of the problems, of course. We don't have an animal model of BV, which is something I think would be really lovely. And they actually showed that when you did that, you increased activated genital CD4 cells in the mice at the cervix. So this story is coming together. It's kind of ragged, lots of pieces that involve mice and small numbers and different approaches, but I think it is becoming a cohesive story. Now I'm going to shift just a tiny bit, and then I'll wrap up by, actually I hope, creating a transition for Sinead to come up and talk about PrEP. Everybody knows that the data on PrEP are fantastic for men. We're already seeing declines, Washington, D.C., probably in part because of treatment as prevention, but also maybe PrEP is seeing dramatic reductions in the incidence of HIV. Great data for men, not such great data for women. And why do we think that happened? We think women are spacey and they don't adhere to PrEP, and I can say that because I'm a woman. Is that really the case? Okay, we have blamed the lack of PrEP efficacy, both oral and vaginal, with tenofovir-based products, okay, on adherence, and even the dapivirine vaginal ring. We've blamed the less effective finding in young women on low adherence. So I think the question is now, is it all non-adherence? Clearly in the study that I was involved in the FemPrep study, and in the FACS-001 study that Sinead was involved with vaginal tenofovir, adherence was a huge problem, and it did mediate a lot of the lack of efficacy. But there are some very intriguing data coming out. First of all, we know that the rectum is not the vagina, in case you didn't know that. I'm happy to tell you that definitively. I don't have imposter syndrome when I say that. But it's less, part of the challenge is that it probably metabolizes tenofovir differently. So tenofovir is simply not as activated and concentrated in endocervical T cells or endocervical cells as it is in the rectum. So again, a biological disadvantage from women, and that's the Cottrell paper, which I can talk about separately. I didn't want to take the time. So does the vaginal environment play a role? Well, this is Nikki Klatt's paper, which just came out in Science literally a couple of weeks ago. And what this is the publication that followed on the Durban IAS meeting. And they, again, used bacterial profiling by proteomics or mass spectrometry in the 688 women from the Caprisa 004 trial. And again, you're seeing here that distribution of the community. And they then went in, again, to take a look at the cumulative HIV infection probability by treatment assignment in women with vaginal lacto versus non-lacto bacterial dominance. And what you can see here is that the probability of HIV infection in the placebos versus the controls in the lactobacillus dominant group on the left did separate out. It was clearly different. So tenofovir worked in the women who were lactobacillus dominant. In the women who were non-lactobacillus dominant, tenofovir did not work at all. Now, what's the problem with this? To me, the biggest problem is lactobacillus dominant just lumps everybody together. So there is no distinction between inners and crispidus. And when you think about Doug Kwan's paper that showed that dramatic difference in HIV incidence for community state type 1 versus 2, I think that's a big problem. Nonetheless, they still showed this marked distinction in tenofovir efficacy by whether these women had lactobacillus or no lactobacillus. And that, I think, was pretty compelling, okay? They then went a little bit further and looked at the metabolism of tenofovir by Gardnerella vaginalis and BV-associated bacteria. This has gotten a lot of press. The bottom line, I won't take you through these figures in the interest of time, but the bottom line is that they added Gardnerella vaginalis, Adipobium vaginae, and Lactobacillus crispidus, and I think Lactobacillus inners, if I'm not mistaken, yes, two in vitro cultures systems of Gardnerella vaginalis, and essentially showed that with tenofovir in place, the metabolites of tenofovir were effectively gone, or tenofovir, functional tenofovir, was taken out when Gardnerella was there. So this has given rise to this concept that Gardnerella vaginalis essentially chews up tenofovir, and maybe the women in the trial who were lactobacillus non-dominant had a lot of Gardnerella, and that was chewing up whatever tenofovir they were using. Maybe they weren't that adherent, so even if they had been protected through partial adherence, then they were out of luck because their Gardnerella was the bad boy, and it is a bad boy. So what does that really mean? How should we put this together? Well, I think you can say from these data, fascinatingly, that tenofovir gel efficacy was threefold higher in women with this with lactobacillus dominance and not effective in those who did not have lactobacillus and were dominated by Gardnerella. So I think the vaginal microbiome probably interacts with some topical antiretrovirals. We don't know what that really means or what that's about. I think the Clapp paper has some great hypotheses, but there could be other things going on here too. I already mentioned the PK/PD, pharmacokinetics, pharmacodynamics of the rectum versus the cervicovaginal environment. Unfortunately, that's different. However, beautiful data from the Partners in Prevention study that Renee Heffron presented at CROI, saying it really doesn't matter if you take oral PrEP. So you should be fine. If you're adherent to your oral PrEP, you're going to overcome these problems. At least that's what they saw. Nikki Klatt did finish her paper, though, by saying vaginal pH screening and microbiota monitoring and treatment are potential adjunct interventions to enhance PrEP impact in women. Well, what are we going to do? We're going to screen and monitor, and what? Are we going to treat them? Are we going to give them lactobacillus? That wasn't really addressed. So I'll just point out a really, I think, promising approach. This is Jen Balkus's paper, again from last year, that presented the data from the Preventing Vaginal Infection study. And if you may recall, Scott McClellan's group showed previously that giving women periodic presumptive treatment for vaginitis, namely monthly metronidazole and an antifungal cream, reduced, not surprisingly, the incidence of vaginal candidiasis and BV. What this group then went on to do was to look at whether that reduction in BV was associated with a reduced risk of acquisition of sexually transmitted infections. So let's put our hypothesis to the test, right? And indeed, small numbers, but what Jen and colleagues showed was that if you combine the outcomes of chlamydia, gonorrhea, and mycoplasma genitalium, you showed a significant reduction in acquisition of STIs. Small sample size, but clearly a very interesting trend that should really, I think, re-energize those of us who are working hard and care about maintaining a normal vaginal environment. So I am basically finished with 35 seconds left. So just to wrap this up, how would I conclude? I think clearly a Lactobacillus crispidus dominant microbiome is associated with a lot of good things, and especially a lower prevalence and incidence of BV. I do think we need a lot more work in this area. We really are just starting to pull these threads together, and I think maintaining this environment, the normal environment, is really going to be the holy grail. But we desperately need new interventions, right? I mean, what do we have going on? I love this cartoon. Actually, I got some pretty good ideas when I was in the box. The box is a safe place, especially when you're talking about vaginal microbiome. It's a safe place, right? We want to think inside the box. We want to think about new antibiotics. Maybe we want to do a new version of metronidazole, right? Maybe we want to do a new version of whatever. I don't think so. What do we need to do? We can't solve our problems with the same thinking we used when we created them. Kevin DeCock said this last night, using different words. And I think when we think about what we should do with the vaginal microbiome, there are host of things we need to start being more creative about. I think creative approaches to partner management, Kat Bradshaw and I have talked a lot about doing a study involving topical and oral therapy for males. I think not having topical therapy probably just doesn't make much sense to me. Modifying the hormonal environment. There's lots of ways we could do that. We're doing a study now in Kenya, and Ken Nguri is here, going to give you an update on our study looking at NuvaRing to modify the vaginal environment in Kenya. Lactobacillus repletion, I mentioned Craig Cohen's study. Vaginal hygiene interventions, beautiful summary by Nicola and colleagues looking at the risk for vaginal hygiene, incredibly prevalent. Biofilm disruption, new antimicrobials, and menstrual suppression. Menses is very much associated with onset of BV, and we could certainly deal with that with hormonal therapy. So I'll just stop there. I really want to thank IUST for this incredible honor, especially David, who's just been a wonderful, wonderful colleague over the years, and then a lot of people who've given me slides or really talked through some of these issues with me. And I'll stop there, and thank you very much for listening. Beautiful, thank you. Thank you very much. I hope it was what you wanted.
And happy birthday to you in Rio today, Jean. I'm going to hand over to Charlotte now. Thanks, Charlotte.
Thank you, David, and thank you, Jean. Happy birthday. That was a lovely tour de force. I think we all learned a whole lot, and we have a lot to think about. So I'd like to introduce our next speaker this morning. This is Dr. Sinead Delaney-Moretley. She is the Director of Research and an Associate Professor of the Research HIV Institute at the University of Witwatersrand in South Africa. The title of her talk is the implication of the implementation of PrEP. Dr. Delaney-Moretley is the recipient of several awards, and most notably, she is a recipient of the Best Emerging Scientist, Women Scientist, with the South African Department of Science and Technology, and she's received an award from the South African National Research Foundation, and she's an investigator on the FACS 001 multi-centered South African trial of tenofovir. So we're anxious to hear what you have to tell us this morning.
Thanks very much, and good morning, everyone. It's great to be here. I was reflecting when I was listening to Jeannie that I think the last time I came to ISSTDR, it was incredibly eventful because we just had started to hear the results of the PrEP trials. So it's been a while, and it's great to be back, and I like the idea of surfing the wave. Okay, now I'm gonna... Okay, there we go. Oops. Oops. Okay. All right. So this morning what I hope to do is talk to you a little bit about PrEP, the current status, where we are at the moment, and then some of the barriers to implementation, but what I also think are potentially opportunities for us, particularly in the sexual health field, and then spend a little bit of time looking to the future. Oops. So some of you may recall that in November 2015, the WHO recommended that oral PrEP be provided to individuals at substantial risk for HIV infection as an additional choice in a package of combination HIV prevention approaches. And that recommendation was a strong recommendation with high quality evidence from several trials. In fact, the recommendation was based on key evidence from a systematic review involving data from 12 randomized controlled trials of oral PrEP effectiveness that had been conducted in a range of populations across the world, including many who have sex with men, serodiscordant couples, heterosexual women, and injecting drug users. What they found in the systematic review was that PrEP was effective in reducing HIV risk across age, gender, ARV regimen, and mode of HIV acquisition. And that there was no difference in the frequency of adverse events between those that had received tenofovir or TDF-FTC and those that had received placebo. Importantly, and what had been a big concern prior to these trials, was that resistance overall was low, and where it was observed in people with acute HIV infection who had initiated PrEP. Another important observation from this meta-analysis was that there was no evidence for sexual behavior change across the groups using either TDF, FTC, or compared to placebo. So this recommendation was really powerful, but it did come with some caveats, and the most important of those was that effectiveness really does depend on adherence. And what we saw is that in trials where they observed greater than 80% adherence, effectiveness was as much as 70%. So you can see up at the top, those trials where adherence as measured on blood levels was high, we also saw high levels of effectiveness. But there were two trials that you heard mentioned a little bit earlier. Whoops. The FemPrep and the VOICE trial, both trials conducted in women, where we saw much lower levels of efficacy, and this was found to be associated with a low level of adherence. When tested with tenofovir plasma levels, plasma detection was less than 30%, suggesting that women had been poorly adherent to tenofovir. But as Jeannie mentioned, the question arose, was it just behavioral, or was there potentially some biological explanation for why we saw this lower efficacy of PrEP in women? And this is data that comes from a PK/PD modeling exercise using mucosal samples collected from 47 healthy women, which was used really to predict the relationship between drug concentrations and the potential for efficacy. And what you can see in the top panel is data from rectal mucosa, and in the bottom panel is are data from samples collected in the female genital tract. And importantly, what we can see is that a couple of things. The first is that the overall about 98% of participants in the study had achieved some level of protection with tenofovir by day 3, which is an important observation given some of the debate about how soon is the time to protection. But the second striking thing you'll see is that while with in the rectal mucosa it required very few doses, you can see on the bottom the number of doses per week. So you can see here with 2 doses you have almost complete protection, compared to in the female genital tract where it required 6 to 7 doses per week to achieve the same level of protection of the female genital tract, suggesting that there is a higher bar for adherence to women when it comes to protection of the female genital tract as opposed to the rectum. So these guidelines have been incredibly important for helping us to anticipate how PrEP might be effective. And modeling studies have suggested that PrEP is likely to have the most impact if it's offered to populations which have an incidence of more than 3% per 100 years. When we think about who has that high rate of new infections, the map highlights areas of the globe where we see populations with high rates of new HIV infection by mode of transmission. So in many parts of the world, key populations would benefit from PrEP. And frequently these are populations that already have poor access to HIV prevention services while having high-risk behavior. But strikingly, in sub-Saharan Africa, adolescent girls and young women are also an important population for inclusion in a PrEP program, as might serodiscordant couples in which the HIV status of one of the partners or there is a positive partner as well as a negative partner. Oh. So I apologize. So as a result of these recommendations, we've seen since 2010, when the first trial results became available, that the registration by the FDA of Truvada for PrEP in 2012, followed by recommendations from both the CDC and WHO in 2014. And then following these most recent recommendations by WHO, a rapid move to register Truvada as PrEP in over 17 countries, including some countries in the global south. So just how are we doing then with implementation of PrEP post-licensure? This is data that has been collected from US retail pharmacies in the period between 2012 and 2015 and was presented by the team from Gilead Sciences at the IAS meeting last year in Durban. And what they essentially showed is a massive increase in prescribing for PrEP in that period, a 738% increase over the period. They also, in this analysis, looked at factors that were associated with prescribing and showed that generally men, people over the age of 25, and certain states, there was far higher rates of prescribing. But importantly, health disparities in access to HIV prevention still persist, and there was much lower frequency of prescribing in women, in people under the age of 25, and people actually living in states which had a high burden of HIV infection. Interestingly, in June 2016, last year, we saw South Africa include PrEP in its public-funded HIV prevention program. South Africa was one of the first of the countries in sub-Saharan Africa to include PrEP in its program, and there is an image of the health minister announcing the program, which meant that PrEP would be offered to 3,000 sex workers at 10 sites across the country. What is noteworthy, in fact, is that prior to this announcement, mathematical modeling had shown that whilst PrEP to sex workers might be of some value, because of the higher prevalence of HIV in this community, in fact, there might be very little public or population level impact on HIV. And the population that really would benefit from PrEP would be young women aged 15 to 19. Mathematical modeling, supported, I think, by various epidemiological and phylogenetic studies, supports the idea that young women become infected at very young ages in South Africa, and that PrEP would allow them to protect themselves against infection for a long period of time, which would, in addition, prevent them transmitting infection to their partners during a long period of sexual activity before they perhaps settle down with a steady partner. We've also seen, literally in the last month, a number of updates suggesting that this expansion of PrEP is going beyond the US and parts of sub-Saharan Africa. Last month, we saw an announcement that Belgium, Portugal, and Brazil would provide PrEP through their health services, and Morocco had announced a PrEP study. And then a couple of weeks ago, or just last week, we heard that the NHS, after a long battle, in fact, would include PrEP in its program for HIV prevention. And there was a further announcement this week that the FDA was about to approve generic Truvada for use as PrEP. So an enormous amount of movement in the field in the last 18 months. And what has that really meant in terms of numbers of people on PrEP? Well, according to colleagues at UNAIDS, there are about 160,000 people on PrEP. But in the UNAIDS 2016 report, the UNAIDS set a target, their Prevention 2020 target, of 3 million people on PrEP by the year 2020. So the proportion of people on PrEP at the moment is less than 5% of the target that we have. And so there is still an enormous amount of work to do. And some of what we need to reflect on are what are the barriers to implementation of PrEP. So I thought it might be useful to think of another disruptive technology that changed the world about 60 years ago. About 60 years ago, the oral contraceptive pill was licensed by the FDA for use in women for the prevention of pregnancy. And like PrEP, it was life-changing. In fact, I will come to talk about it as a disruptive technology. If you look at the comparison between the two products, initially PrEP was approved for treatment, while oral contraceptive pills were actually approved for the treatment of menstrual disorders before they were developed as contraceptives. What both of them offer, though, is individual control over HIV over prevention. And 60 years ago, the same concerns that we have for PrEP were the concerns that we had for the oral contraceptive pill: safety, cost, uptake, adherence, and potential for changes in sexual behavior. So why should we consider these disruptive technologies? Well, a disruptive technology is one that displaces an established technology and shakes up an industry or creates a completely new industry. Often, when we see disruptive technologies, they lack refinement. They often have performance problems the first time around. And because they're new, they appeal to a limited audience, but sometimes they actually attract a new audience who might not previously have used the technology. They may be initially dismissed by the mainstream because they don't reinforce current mainstream goals or beliefs, only to lead to the mainstream being blindsided as the technology matures and gains a larger audience and a market share or threatens the status quo. So while this is an example from a business administration book, I've heard this analogy being used in describing vaccines, and I actually think it's also very useful in describing or thinking about some of these reproductive technologies. Because the important thing about disruptive technologies is that we need to consider that they might not be perfect, that they might need to fail a couple of times, but ultimately they will be game changers because, in part, they respond to a desire that a new group of users might have. So I'd like to spend a little bit of time thinking about what are some of the barriers to PrEP implementation, and could some of that be because PrEP does not fit into the mainstream view of HIV prevention, and does using a disruptive technology framework help us to think about ways to advance PrEP development and delivery to those that need it. So what does PrEP offer? Unlike condoms, which are the available method of HIV prevention, we have evidence that PrEP decreases anxiety, it increases communication and disclosure, it increases intimacy and trust, a sense of community, self-efficacy, sexual pleasure, and intimacy. These are all characteristics that you don't hear when describing the condom. So in some ways, what we argue is that PrEP may be appealing to a market that hasn't necessarily used condoms. This is a quote from a PrEP acceptor at my clinic in Johannesburg, and she said, "My previous sexual experiences were the main motivation for joining the studies. I had a series of unprotected sex and waking up the next day regretting it. I was very excited about PrEP because of the idea that I would take control of my future and own my health." And here you see PrEP as an empowering technology. So importantly, PrEP is not for everyone and not forever. And we need to recognize that when talking about PrEP and thinking about PrEP uptake, importantly, in many parts of the world, when we're looking at PrEP uptake, we need to understand that it may be taking place in the context of a lack of knowledge and access to PrEP. And both are incredibly important for driving demand. And in some open-label studies, we've seen that people may not necessarily accept PrEP even if it's offered to them, because the prevention options that they have actually do work for them. But importantly, stigma is a key barrier to PrEP use. There's stigma around the use of antiretrovirals, that you may be identified as being HIV positive. There is stigma around the potential that you could be identified as being HIV positive. And there's also stigma about being at risk for HIV, and particularly with young women or young people, the fact that you might just be sexually active. So important gatekeepers in the delivery and implementation of PrEP are prescribers, and frequently they are the ones who may influence and have been shown to influence people's uptake of PrEP. So I'd like to spend the next section of this talk really reflecting in part on my own experience as someone working in South Africa who has worked with policymakers and prescribers around the development of a PrEP policy and come to understand some of the anxieties that the mainstream may have about PrEP and potentially to try and address them in a series of considerations. So what are the concerns and barriers to implementation? Well, the first thing that people have worried about, although perhaps less so now, is safety. And the reason is that this is a drug that we're offering to essentially healthy people, and so even the slightest side effects may be more consequential than someone who needs PrEP for treatment. In addition, we're offering this drug to women, particularly of childbearing age, and that may generate another set of anxieties. But the data to date suggests that PrEP is well tolerated and that the most common side effects that are observed with PrEP initiation are either nausea or a GI upset or headache. Generally, 1 in 10 people experience that, but those symptoms resolve within about a month. The other concerns related to the effects of tenofovir on the kidney. Tenofovir is metabolized by the kidney, and because of that, there have been concerns about the renal safety of tenofovir. But importantly, in the trials, what we saw was less than 1% of grade 2 to 4 elevations of creatinine in the participants who were in the trials. These elevations were transient and we saw return to normalization with cessation of drug. Similarly, we see very little loss of bone mineral density, no increased risk of fracture, and a return to baseline upon withdrawal. Resistance was also a concern. We've identified that that mainly occurs during the acute period of infection, and so steps have been put in place to address frequent testing and identification of acute infection. But importantly, these studies were done in selected populations, and really we need longer-term follow-up in diverse populations. And ultimately, what we need are the development of safer drugs and formulations, as we saw with contraception. Another important issue, particularly in South Africa, has been a concern about the safety in breastfeeding and pregnancy. A recent paper was published by Lynn Moffinson reviewing 33 studies that derived data from women who are HIV infected or uninfected. And essentially what we saw were no increase in birth defects or poor pregnancy outcomes or low birth weight or influence on infant growth, which is very reassuring. We also saw no increase in maternal mortality. And another study done by the Partners PrEP team showed that there were negligible amounts of tenofovir detected or transmitted through breast milk. So all of this has led to the recommendation by the WHO that a conversation be had with a woman who is pregnant to discuss the risks and benefits of taking PrEP during pregnancy. This has important implications for countries which have nurse-led systems where they may not necessarily be able to make these decisions about risks and benefits, and it may require clinician involvement. And this is one of the reasons for the delay in implementation in certain countries. But whatever we do choose to do, we will need to continue active surveillance of maternal and infant outcomes for PrEP programs. So the second barrier that people have talked about, in the same way that they talked about it with contraception, is the issue of cost. So the first thing is that cost can place an enormous logistical and economic burden on the user of PrEP themselves to return for clinic visits and to get prescriptions. And if there isn't public funding, this can be an enormous barrier to PrEP use. The other issue is the cost to the health system. So while there have been many studies that have showed that PrEP will be cost effective, particularly if delivered to people at high risk for HIV infection, there are questions of affordability. These two pie charts show the breakdown of costs per client year for South Africa when considering PrEP for adolescent girls and young women. And what you can see is that while drug is a substantial portion of the cost, 3/4 of the cost is also related to the service delivery. And so that's a really important cost for the health service and will require significant investments in the health system, which may be an even bigger barrier. And we saw, I'm really sorry for this. We saw this play out this week in South Africa with discussions in newspapers and on listservs about the PrEP targets within our National Strategic Plan, and activists arguing that we are falling way behind our HIV prevention goals because of these low targets. But in part, the reason for these low targets is the extreme concern around whether or not the Department of Health and the health system actually has the ability to deliver PrEP to the people that need it. So cost considerations go way beyond the price of a drug through to the costs associated with ailing health systems. Now, an even bigger concern is the risk compensation. But is this a reason not to act? So there are many concerns that have been raised in relation to PrEP, and we've talked a little bit about them because they are framed in terms of being perceived as a threat to consistent condom use, to potentially undermining condom use and the gains of condom use, a relatively cheap technology. There have also been concerns about the medicalization of prevention and the idea that we will just use a pill to prevent HIV instead of addressing the underlying social and structural barriers to HIV prevention, particularly for young women and key populations. And in some circles, there have been concerns around, in sex worker circles, that PrEP could actually be mandated and sex workers may be forced to use PrEP, ignoring the kind of human rights framework that's been set up around HIV prevention for sex workers. And I think all of this is very well summed up in terms of the moral panic that goes with PrEP, the idea that it might be associated with a loss of sexual restraint, very much akin to what we saw when the oral contraceptive pill was introduced 60 years ago. So what does the data tell us? Well, when we look at the data from the trials, we see that there was no increase in unprotected sex in the trials. So before and after a particular time point, we see no difference in unprotected sex in serodiscordant couples or STIs or pregnancy after the time when placebo was stopped. When we look at data from the PROUD study, the PROUD population was at considerable risk for HIV and had high rates of STIs in the year preceding their enrollment in the trial. And what we also saw is that although they had high rates of STIs, incidence didn't vary between the deferred and the immediate PrEP arm. And all of this needed to be interpreted against the background of rising STI rates in Europe. So one of the additional concerns is, do STIs potentially reduce PrEP efficacy? And you heard Jeannie talk a little bit about the BV story. But just to confirm that there's very little evidence to indicate that STIs or co-infection with an STI might lower PrEP efficacy, which would be a particular concern. There's some data from the IPREC study which showed an incidence of syphilis of around 7%, but no difference by arm. And similarly, similar data from the Partners PrEP study. And as I mentioned, also in Partners PrEP, Renee Heffron presented data showing no effects of oral tenofovir on BV rates or BV rates on oral tenofovir. So to date, we also haven't seen evidence from open label studies. There were high rates of STIs throughout these trials, and they were impressively high and need to be responded to, but are not necessarily the cause of PrEP. Another interesting example comes from the US, where an analysis of data from Kaiser Permanente showed this disconnect, in fact, between incidence of HIV and incidence of STIs. In the bar graph, you can see high rates of STIs observed in a 12-month period, but in fact no HIV incidence. So people may have been taking their PrEP well, and what we really need to hone in on now is what additional do we need to do to reduce rising rates of STIs. And what are the implications of this potential either high background or increased rates of detection or potentially increased rate of STIs that we might see in low and middle income countries, which already have a higher background of and burden of STIs. So what I want to suggest, in fact, is that when we see some of these issues, they are opportunities. That PrEP is a new sexual health technology, and it provides opportunities for us in sexual health services to strengthen sexual health services in both high and low and middle income countries. But also it's a prompt to develop longer acting, less user dependent products that can potentially be combined with contraception or STI treatment. And these we would call multi-purpose technologies. So when we think about strengthening services, there are a number of key questions for implementation for PrEP, which may in fact be worth considering when we consider the model of delivery. So the first question is, how do you create demand? What information is needed? What testing services are needed? How can you help people to identify their risk? How can you draw them into services without necessarily stigmatizing them? And how do you support supply? What do services need? What models of delivery are needed? Are they fixed clinics, mobile clinics, outreach services? Can we look at alternative models like sexual health services, family planning clinics? And how do we integrate with other interventions? And this is, I think, a question for the STI world. And then how do we support effective use? And what does adherence support look like? So some of the innovations that we've seen for PrEP have been counseling, SMS or text reminders, clubs and drug feedback levels, as well as targeted intensification for those that need more support. So these are a number of innovations that are being considered for PrEP, but could they also be leveraged for STIs? Do we believe that PrEP programs could reduce STIs? Well, probably yes, because we see people for PrEP visits quarterly at the moment, and they come for regular HIV testing and could be offered STI testing. It's an opportunity to screen and identify people for STIs and also ensure partner notification. And we may be able to add on additional services to PrEP services, like the addition of PEP in PrEP users, STI PEP that is, but also the potential role of self-testing kits, not just for HIV, but also for STIs. So I wanted to give you a couple of examples of where we're starting to see innovation in service delivery. This is New York City's public health approach to ending the HIV epidemic. Essentially, they've used epidemiological data to inform and transform their sexual health clinics to make them destination clinics for people in the city. They've also launched PrEP and repaired their PEP program so that PEP can be an entry point for PrEP. Their clinics are branded as status neutral. You test, you can get a treatment, and it doesn't matter whether you're HIV negative or positive. There's something for everyone. And they have a number of novel delivery strategies and novel interventions, such as their Play Sure kit. Very briefly, I'll touch on the idea of PEP with PrEP. This was a study nested in the IPIGAY study, where men who were randomized either to on-demand PEP doxycycline 24 hours after sex or no PEP. And what we saw in the combined results, that people who received the PEP, there was some potential benefit in terms of time to first STI. This was seen most with syphilis and chlamydia, and no effect was seen for gonorrhea. So very briefly, I'll talk a little bit about, those are examples done in high-income countries, but what can we do in the global south? This is a clinic in Johannesburg where I work. It is a youth-friendly service. It offers comprehensive services for HIV testing, treatment, PrEP, STI treatment, and family planning. Young people aged 10 to 24 have an HIV prevalence when they attend testing services at that clinic, and we see about 1,000 people a month. And one of the things that we're evaluating at the moment is whether it's possible to include PrEP in a combination HIV prevention package, which also includes screening for gender-based violence. So this gives you an idea of how participants enter the study. But more importantly, I wanted to show you were the tools that we've introduced, which include risk score assessments and screening for STIs, a film and materials that talk to young people, and it's on YouTube if you'd like to get it. But ways basically to assess people's risk, which may also be valuable for STI screening. We also involve stakeholders and communities in engagement around HIV prevention and have clubs that support adherence. But these can also be used for other activities. So to date, we've enrolled 320 PrEP users, and we're learning a lot about PrEP delivery to adolescent girls and young women in sub-Saharan Africa. But importantly, a cautionary note. This is STI data from those young women who were screened at our clinic, and what you can observe are the very high rates of chlamydia and gonorrhea in a population aged 16 to 24 years old, and about 20% were also HSV2 seropositive. So it's clear that PrEP services really do need to think about STI control as well. So what does the future hold? Well, let's go back to the lessons from contraception. If we add more methods into the mix, we're likely to increase total coverage. This is what the future looks like for PrEP: rings, injectables, broadly neutralizing antibodies, and other formulations of Truvada, as well as vaccines. Most of these are in phase 2 or 3 trials, and we're likely to see results in the next 4 to 5 years. And further down the line, we have implants, some of which may be combined with an antiretroviral, so that we could begin to see combinations of products. And perhaps we should also think about combinations of products like a therapeutic HSV vaccine shot with an injectable. So in conclusion, I think we've made enormous progress in the development of a new biomedical technology that has the potential to be a game changer for HIV prevention if it's deployed correctly. The concerns about PrEP highlight the disruptive nature of PrEP, and these concerns that can be managed as we have done so with other innovations like contraception or even the HPV vaccine. These are concerns that reflect the times we live in, the cost constraints, but also the moral angst that we have about uncontrolled sexual activity. For us in the sexual health world, many of these challenges are actually opportunities to revitalize and re-energize sexual health services, particularly in sub-Saharan Africa. We are going to need new technologies that will improve the performance characteristics of PrEP, and we should see this as a vanguard product. But finally, multi-purpose technologies, which are the future, are urgently needed really to address the multiple burdens of diseases in key populations. I'd just like to acknowledge the people who contributed to these slides.
We have an announcement for those of you who are staying for the last plenary. If you would like to have the translation earphones, they've been passing them out, but just hold up your hands as the translating earphones are being passed out while we get ready for the next speaker, who will speak in Portuguese, although the slides will be in English. So, okay, so our next speaker is Dr. Suzanne Suera. She's previously been the director of the Department of Science, Technology, and Strategic Inputs in Brazil. But in 2009, she was named by the World Health Organization and PAHO for the post of Regional Advisor on Sexual and Reproductive Health. And then, in 2014, she was nominated to be the Chief Director of the Latin America Center for the Perinatally Unit of Women and Reproductive Health. So the title of her talk today is Maternal to Child Syphilis Transmission in Latin America. So welcome, and if we have time at the end, we'll have our 3 speakers for questions, but it will depend upon the time. Thank you.
And if I could make a quick announcement, we are trying to get more headphones. They are coming. I apologize for your hands being up. And then just a note that there is a conflict. The Lancet Infectious Disease Commission will not be in Segovia room. It will be in the Alhambra room, because there are conflicting schedules. So Alhambra room for Lancet ID.
Yes. Opportunity. So also to ask for your colleague, you have many Brazilian, that side of you can give you the translate machine. So, thank you. Então, vamos falar português nessa conferência. E talvez algum espanhol, porque como eu vivo no Uruguai, o meu português está cheio de espanhol. E vamos começar de uma maneira mais tradicional. Em nome da Organização Pan-Americana de Saúde, eu quero agradecer o convite para apresentar essa conferência e, pessoalmente, agradecer a duas pessoas que tornaram possível minha presença aqui, me ajudando, a Angélica Miranda, que é a diretora científica desse congresso, e a Mariângela Freitas, que é uma assessora em tempo parcial do Centro Latino-Americano de Perinatologia. Mas, antes de começar, eu queria a licença poética dos que não são brasileiros para falar desse sentimento que é estar no Brasil, para nós que vivemos no estrangeiro, e que eu cheguei ontem à noite justinho na hora do hino. Estava começando, eu cheguei, consegui sentar, e começou o hino. E o que a gente viu? A gente viu um karaokê desafinado, né? A música estava num ritmo, nós estávamos cantando a letra e a letra vinha depois. O karaokê estava desafinado, mas os brasileiros não desafinaram. E foi tão bonito, e eu falei, nossa, isso é uma recepção. A gente chega, o karaokê pode desafinar, os brasileiros não vão desafinar no hino nunca. Muito obrigada pela recepção afinada do hino. O primeiro quadro que eu vou mostrar para vocês, ele é um quadro que podia ser até histórico, mas não é. Ele é um mapa que a gente encontra no sítio da Organização Mundial de Saúde, e ele traz o número de grávidas positivas para sífilis. A primeira coisa que salta aos olhos é que ele ocupa o hemisfério sul. Quase é como se o norte não fizesse parte dessa infecção. A segunda coisa que nos causa, não espanto, porque nós escutamos ontem, escutamos hoje e em todas as reuniões anteriores, que a sífilis continua aumentando em muitos lugares. Como vocês sabem, um indicador sentinela da sífilis é o número de embaraçadas positivas para sífilis. Então, a gente vê que esse número não diminuiu. E a outra coisa que também ontem foi dita é que nós estamos falando de uma doença que é histórica. Há mais de um século nós falamos de sífilis. Nós conhecemos a tecnologia para diagnóstico, nós conhecemos o tratamento, não nós da área de saúde, não a gente que é militante, mas o Banco Mundial diz que é uma das intervenções mais custo-efetiva. Então, por que nós seguimos com sífilis? Porque temos sífilis e sífilis congênita. A resposta não é tão cartesiana, não é verdade? A resposta não é simplesmente ter um tratamento e ter um diagnóstico que garante a eliminação. Não é assim, tão cartesiano, tão simples. A resposta seguramente está fora dessa lógica tão, talvez, médica ou de saúde pública, e vai de encontro aos determinantes sociais. E uma palavra que eu acho que está muito hoje em discussão, que é a equidade. Hoje, a partir do momento que nós assumimos um novo marco global, com os Objetivos de Desenvolvimento Sustentável, né? Não sei quem não conhece, é esse símbolo. Às vezes eu visto ele e algumas pessoas perguntam se está relacionado com os outros movimentos. Eu digo, não, aqui tem mais cores, 17. E nós estamos buscando justamente ampliar, sair do campo tão restrito que é o campo puro da saúde, para entender que várias das nossas intervenções falham. Falham por falta de conexão, de intersetorialidade, de visões mais amplas. Muito bem, esse é o todo mundo sabe as taxas, elas foram amplamente discutidas, e o que a gente vem vendo na América Latina e o Caribe, que é o tema dessa apresentação hoje. Então, também, todos vocês sabem, a Organização Pan-Americana de Saúde, ela é a secretaria técnica dos países. Portanto, responsável... Isso daqui dá um trabalhinho, né? Eu vou passar de novo. Portanto, responsável por coordenar os planos e as resoluções. E olhem vocês, há 20 anos existem vários documentos, resoluções que são assinados pelos ministros de cada país. Ou seja, a Organização Pan-Americana de Saúde, a OPS, a PARRO, vem colecionando iniciativas há mais de 20 anos, e eu vou me deter na última delas e na penúltima. A última, lançada no passado, ela é uma ampla resolução para eliminação, para diminuição, para o controle, para a prevenção das doenças infecciosas, transmitidas por infecção, TST, e de HIV. Mas, em 2010, nós tínhamos conseguido um marco importante, que era discutir a eliminação dual. Ele vai rápido, aí ele não vai. Vamos lá. Então, um trabalhinho, mas a gente volta. Tecnologia é isso. Funciona e, às vezes, não funciona. Aqui. Nós decidimos por uma iniciativa regional, por primeira vez dual, para sífilis e HIV. A ideia era, de fato, promover, em quase todos os países, um novo paradigma para a eliminação disso. Do ponto de vista do nosso secretariado técnico, nós preparamos um conjunto de marcos. Todas as guias que vocês veem em cima são guias que vão do mais político, desde como fazer advocacy, a fazer monitoramento, como fazer pesquisa em mulheres embaraçadas, no puerpério, para ver sífilis, que é um ótimo indicador. E embaixo vocês veem uma série de estudos e informes que tanto foram produzidos no centro, que eu sou diretora, como nos próprios países. A gente buscava o que pensávamos que seria... Está um pouquinho ruim isso. Eu vou pedir auxílio para o pessoal que está atrás. Isso, eu estou vendo. Volta uma, por favor. E eu peço para... Pronto, perfeito. Então, quando vocês olham esse slide, eu me lembrei da frase do nosso conferencista ontem: pode parecer simples, mas não vai ser fácil. Qual era a visão principal? Começando daqui. A visão é que a gente quer um mundo mais justo, com gerações livres de HIV ou de sífilis congênita. Essa sobe? Esse. Como é... O que a gente espera disso? A gente esperava ter uma transmissão de HIV menor do que 0,2%, menor do que 2%, e menos de meio casos, menos de 0,5 casos a cada 1.000 nascidos vivos de sífilis congênita. E isso incluía os natimortos, que é uma das discussões que a gente vai ter hoje aqui. Se vocês olham a coluna lilás, ela toda pede que a gente discuta as ações programáticas e pede que 95% das mulheres tenham acesso ao pré-natal a um parto institucional. Parece simples, mas nada fácil. Que elas tenham um diagnóstico e um tratamento, que os serviços sejam integrados, que funcione a informação e que a gente possa ter, com isso, uma prevenção e uma promoção mais eficiente. Quando a gente olha as linhas de ação, a gente vê que existe uma complicação aí relativa ao que a gente chama hoje de serviços fortes ou resilientes. Como operam os sistemas de saúde? Como vai o mercado de saúde? Do que nós estamos falando quando a gente fala de integrar serviços maternos com serviços de prevenção de DST e de AIDS. Todos esses elementos foram discutidos durante meses, estão nas publicações, e levavam a um entorno que nós consideramos que era o mais importante. Isso está claro, está no verdezinho claro embaixo, mas eu acho que é a base da discussão que eu inicialmente coloquei. Partia de princípios, como a discussão de direitos humanos, de gênero, de equidade e de populações vulneráveis. E é aí que está o difícil. Muito bem. A cada ano, a organização faz uma avaliação de como os países estão respondendo. Para isso, nós estabelecemos um conjunto de elementos que fazem parte dessa avaliação. O primeiro elemento é se o país fez uma adesão ou não ao plano, se ele faz parte disso, se ele, de alguma maneira, tem no país alguma decisão de como ele vai proceder em relação a isso. Apesar de que a gente tinha um plano regional, os países da nossa região são extremamente diversos e, naturalmente, a cada um deles cabia ter autonomia para responder isso. Nós também, a maioria dos países tem um plano. Nós perguntamos como funcionava o sistema de monitoramento. Não foi surpresa descobrir que, para HIV, nós tínhamos sistemas mais eficientes do que para sífilis. Muitas vezes, em sífilis, o dado existe e ele não está nem na área materna sendo analisado e nem na área de DST, que analisa outras prioridades. Então, sistemas específicos para sífilis, nem a gente recomenda e eles não são estruturais nos países como o de HIV. E começam aí as dificuldades. Mais ou menos 1,5 milhão das mulheres não têm acesso ao número mínimo recomendado de 4 consultas. E um número importante, como a gente vai ver, não tem acesso ao teste. Então, vamos olhar esse conjunto de elementos e vamos ver, eu vou passar pelos de HIV, que eu não vou ter tempo de discutir, nós estamos discutindo sífilis, nós vamos ver que a maioria dos países tem planos, mas o teste rápido, que é a estratégia, a tecnologia, o elemento definidor para mudar isso, só estão em 19 países, e o sistema de vigilância em 39. Além disso, nós temos problemas conceituais. Todos nós sabemos que um dos desfechos mais temidos de uma sífilis congênita é o natimorto. Portanto, ele faz parte do conceito. A eliminação é 0,5 casos a cada 1.000, incluindo natimortos. Mas, pelo menos, 11 países não têm essa definição, e muitas vezes, quando a gente pergunta, não é porque eles estão em desacordo, é porque eles não têm nenhum sistema de avaliação de natimortos. Quando a gente olha os indicadores, para ter uma ideia dos números, existem 15 milhões de grávidas na região. Eu estou excluindo Canadá e Estados Unidos dessa análise. Então, nós vamos ver a primeira... E aí está comparado HIV e sífilis também para dar alguma dimensão para vocês. Mas quando a gente olha o número de grávidas que não tem nenhuma consulta, ou tem até uma consulta, isso pode chegar a 500 mil. Não é um número pequeno, é um número bem razoável. E quando a gente pergunta quantas mulheres não acedem, não têm acesso a um teste, isso pode chegar a 3 milhões. São dessas mulheres que a análise dos determinantes e quem são essas mulheres, desagregar esses dados, que eu vou tentar mostrar um pouquinho para vocês. Estou começando com o macrodado para a gente chegar no mínimo. Dando um exemplo só do percentual de mulheres positivas, a maioria desses dados faz parte do informe. O informe está disponível aqui, em inglês e espanhol, e também está disponível nas nossas páginas. Mostra que a maioria dos países estabilizou o número de mulheres que ele alcança para o tratamento. Isso é um dado que serve para reflexão. Aqui tem vários que servem, né? Primeiro, poucos países a gente tem dados para os 2 anos, mostrando a inconsistência, a fragilidade do sistema de informação. Segundo, eles não conseguem superar o ano anterior. Ora, se eu tenho 80% e eu quero chegar a 100%, eu deveria ter um acréscimo. Na maioria dos países, isso não acontece. O que a gente viu nos últimos anos é que houve uma estabilização. Pode considerar esse número alto, 84%, mas ele não é, porque, novamente, nós estamos deixando sem tratamento adequado mais de 16% da população de embaraçadas. Então, voltando, 500 mil não consegue nem ter pré-natal. E esse é um dado importante dos estudos qualitativos, como a gente vai ver, porque faz muita diferença nos resultados perinatais. 3 milhões não têm acesso ao teste. E pelo menos, vamos dizer, 1,5 milhão não tem acesso ao tratamento adequado. Então, a gente tem números importantes que impedem que aquelas altas coberturas necessárias façam com que as Américas cumpram com a eliminação. Aproximadamente 23 mil recém-nascidos na região têm sífilis congênita. É uma estimação. Porque eu vou mostrar para vocês agora, e vou passar direto para o gráfico, que ele é mais explicativo, o que aconteceu com os casos reportados. Então, outra vez, são dados oficiais reportados para os países para o monitoramento dentro da Organização Pan-Americana de Saúde. E abaixo, em laranja, estão 36 países. Esses 36 países, se vocês olham assim rapidamente, vão dizer, bom, Suzane, todos eles quase alcançaram, né? O que aconteceu? Nós sabemos que aí tem subnotificação, e aí tem 11 países que não contaram os natimortos, e aí tem países que não têm sistema de notificação confiável, sólido, consistente, que a gente possa dizer, esse é o dado real. O Brasil tem um sistema de vigilância para sífilis há muitos anos. Aqui, nessa conferência, eu vejo vários dos meus colegas que sabem muito mais do que eu, os responsáveis pelo sistema. E, de fato, nos últimos anos, sentado aqui na frente um deles, o Disseu, o Brasil fez um esforço enorme para disponibilizar os testes rápidos. A realização do teste rápido, a melhora da vigilância e também a falta de penicilina fez com que, nos últimos anos, o Brasil tivesse um acréscimo impressionante de número de casos. Ontem, conversando com a coordenadora, com a diretora dessa área no Ministério, a Adélia Benzaquem, ela me disse que sim, o número que eu havia lido nos informes de quase 15 mil casos é o que eles estimam. É bom ou é ruim? Eu acho que tem um lado muito bom disso, que, finalmente, a gente está testando mais mulheres. O acesso ao pré-natal no Brasil é mais de 96%. É bom que a gente tenha muito teste rápido? O que aconteceu com a penicilina? Também não dá tempo de discutir aqui, mas, rapidamente, todos tenham, né, em mente que nós estamos tratando de uma droga que a matéria-prima está na mão de 2 países e que não tem interesse comercial. Essa é a grande diferença. Não tem. A penicilina não é uma droga que dá lucro. Então, muitos países começaram a produção nacional dos medicamentos. Se não existe esse compromisso, nós vamos ter falta de abastecimento de maneira permanente. Como eu disse, não dá tempo de discutir aqui, mas ele é um dos pontos para discussões setorizadas que nós devemos fazer. E isso me remete à minha vida anterior, como diretora nacional de Ciência e Tecnologia, onde um dos nossos trabalhos era conseguir que as drogas para as populações pobres e vulneráveis fossem produzidas e não apenas aquelas em que a indústria tem alto percentual de lucro. Quando a gente faz uma análise geral, isso é um quadro só para vocês terem uma ideia, a gente vê que no Caribe muitos países avançaram, alguns que não são do Caribe também, como Cuba, Chile e outros. A maioria do que está no meio dessas colunas ainda não conseguiu, fez avanços, e a maioria desses países você encontra estudos, discussões sobre auditoria, novas maneiras de fazer diagnóstico, diagramas reversos começando com treponêmico, segmento de recém-nascido, várias intervenções buscando melhorar. E em alguns países a gente não tem informação suficiente. A gente tem vários desafios para eliminar a sífilis congênita. Seguramente, o primeiro deles está relacionado com o momento que a mulher começa o pré-natal. Isso nós discutimos longamente em populações vulneráveis, porque justamente as que mais necessitam, as adolescentes, as que vivem na área rural, as que têm menos informação, que têm acesso demorado aos serviços, porque em alguns países da América Latina ainda precisam pagar, elas chegam no pré-natal já quando nós não temos tempo, a partir da 20ª semana. Além disso, fazer o diagnóstico demora, porque uma mulher pode precisar até 6 vezes, de 6 visitas, para conseguir, isso é um artigo ótimo, que foi publicado, mostrando que ela pegava a consulta, depois voltava para marcar o laboratório, voltava para pegar o resultado. Enfim, era uma sequência de demoras. É preciso também a gente tratar o parceiro, e isso é uma das discussões mais complexas para a sífilis. A mulher não tem nenhum sintoma, ela tem um resultado e diz: eu tenho sífilis, você precisa ir ao médico. Se ela consegue que o marido vá, às vezes o centro de saúde não tem elementos ou não consegue dar espaço para isso acontecer. E o que eu já conversei com vocês sobre a falta de penicilina. Nós precisamos continuar trabalhando para a implementação de testes rápidos. Não existe intervenção mais custo-efetiva, e nós estudamos vários lugares, vários países, e os países que, de fato, avançaram, e está aí o exemplo do Brasil, foi o uso do teste rápido, massivo, como faz o Chile, como fazem muitos países. Primeira consulta, teste rápido, positivo é igual a um shot de penicilina, that's it. Acabou, e a mulher já tem, pelo menos, evitada a sífilis congênita. Depois ela trata se ela precisa de uma 2ª ou 3ª dose. Já considerei para vocês várias coisas do que acontece no sistema de vigilância. Nós precisamos integrar ele, porque muitas vezes ele não está na área materna e tampouco está no que seria na área de vigilância das infecções de transmissão sexual, da maneira que devia estar. E fazer as comparações necessárias para incluir, mostrar a necessidade de incluir o desfecho de mortinato. Isso é, talvez, o nosso maior desafio. E por quê? Então, eu vou trazer para vocês agora, rapidamente, algum dos estudos que o Centro trouxe. Todos têm a fonte, e eles estão disponíveis na internet e também na página do CLAP. O primeiro mostra aquilo que eu venho falando. O que acontece quando a gente não tem um pré-natal? Dobra a possibilidade de ter sífilis. Esse primeiro estudo ele foi realizado na Argentina, em 28 maternidades, e teve 170 mil registros. Ou seja, é um estudo de base populacional em um país com ótimos recursos, como é a Argentina. Nós tivemos esses resultados. Se a gravidez não é planejada, isso pode aumentar a possibilidade de sífilis em 25%. E se ela está numa idade extrema ou é múltipla, como a gente vai ver em outro estudo, em quase 20%. Mas o mais importante está relacionado aos resultados neonatais. Primeiro, a morte fetal, né, o natimorto. Segundo, o baixo peso e a prematuridade. Um outro estudo que nós realizamos, esse foi feito no Peru, levantou dados de uma década, de 2000 a 2010, em 37 cidades, com 650 mil registros. Esses estudos eles são feitos através do sistema informático perinatal, que é um sistema muito conhecido na área materna. Eu me atrevo a dizer que aqui talvez menos conhecido, dado que a maioria das pessoas aqui não está exatamente na área materna. Mas o sistema informático perinatal ele é usado em mais de 20 países da nossa região e a gente consegue ter dados com esses números de registro. Como a gente pode ver, pelo menos, e eles têm no Peru 3 áreas que eles definem bem, a costa, a serra e a selva, existem diferenças importantes quando a gente olha educação, quando a gente olha o número de consultas pré-natal, a paridade, o estado civil e a localização da mulher. Na selva ou na floresta, dobra o risco. O último estudo, ele foi um estudo que nós também realizamos através do sistema informático perinatal, e nós recolhemos de janeiro de 2009 a dezembro de 2012, nós tínhamos um banco de dados com 1 milhão de casos, mas selecionamos, porque eram os registros mais completos, e nós usamos várias análises multivariadas nesse estudo, aqui não há tempo de explicar cada um, e chegamos a 370 mil registros. E aí ficou a incidência, só porque muita gente me perguntou, a prevalência foi de 1,1, nós tínhamos 0,4 de natimortos, e ficou claro aí que a idade materna, a presença de anomalias congênitas, e um natimorto prévio fazia grande diferença no resultado sífilis materna e mortinato. Os mortinatos e a sífilis materna são componentes fundamentais quando nós contamos, né, e vários daqui sabem disso, e não conseguimos alcançar o ODM4 em vários países. Eles permanecem dentro do set de indicadores que nós devemos alcançar em 2030 com os ODS, os Objetivos de Desenvolvimento Sustentável. Mas eu não queria terminar com mais notícias, né? E eu falei muito no final de equidade. Tudo que a gente discutiu aqui foi equidade. Se um país oferece para todos, se todos têm acesso, se as mulheres mais vulneráveis que vivem numa selva, ou que são afrodescendentes, ou que são adolescentes, ou que são mães acima de 35 anos, conseguem ter um acompanhamento. Mas nós temos boas notícias. O primeiro país que globalmente conseguiu receber o diploma, né, de ter eliminado a transmissão vertical de sífilis e HIV foi Cuba, que é o país menos desigual da região, que é o país onde os indicadores de saúde têm uma relevância muito importante no sistema político. Então, esse país conseguiu. Existem outros que estão a caminho, na região e fora da região, mas é bem exemplar, e por isso eu deixei ao final dos números mostrar porque Cuba conseguiu. Não quero falar bem de recomendação, mas sempre as pessoas perguntam, e a gente então deveria ter um pré-natal separado para sífilis? O que a gente deve fazer para funcionar o tratamento de sífilis? De nenhuma maneira. Nos países onde nós conseguimos reduzir a sífilis congênita, na atenção primária que isso foi feito. E é aí que nós devemos colocar os nossos esforços. É preciso, sim, ter testes rápidos e segmento. Isso em todos os países. O que falta é o segmento. A mulher é detectada com sífilis e não existe segmento para isso. Sobre os sistemas de saúde, também falei, mas não custa repetir. Nós temos que melhorar a qualidade, a representatividade. Às vezes o dado existe, ele não está no lugar certo e ninguém analisa. E convidar alunos, residentes, para trabalhar sobre os dados. Quando a gente trabalha sobre os dados, e nós publicamos mais de 12 investigações, paper, publicação nos países, a gente viu claramente que melhora a evidência, ajuda o tomador de decisão a saber o que fazer. Um dos exemplos, no estudo que eu não tenho tempo de mostrar, é o do Uruguai, onde a gente conseguiu localizar os bairros, onde errava, onde quebrava a linha de tratamento, a linha de cuidado. Eu acho que isso é muito importante da gente estimular. É preciso, claro, desagregar dados. Se a gente não sabe a idade, de onde ela veio, nós não vamos conseguir. E, por último, se a gente pensa num futuro, e eu gostei muito da última apresentação, que trouxe elementos de saúde materna, onde existe muita resistência para a mudança, uma mudança fundamental para os nossos serviços. E aí, em linha, e é muito importante ler o documento desse novo marco global dos Objetivos de Desenvolvimento Sustentável, é integrar. Se nós não integrarmos os serviços, se nós não somarmos esforços, se nós não fizermos uma potencialização das ações, nós não vamos conseguir alcançar. Então, os serviços de saúde materna e de DST, HIV, necessitam ter uma integração mais apropriada. Nós precisamos também melhorar a qualidade do pré-natal e fortalecer os serviços como um todo. Eu espero que, no futuro, alguém tome esse primeiro slide, chegue aqui e diga: eu vou contar para vocês uma história. Havia no planeta sífilis congênita e agora não tem mais. Muito obrigada.
Before we leave, there are several important announcements. First of all, please don't forget to hand in your translation microphones as you leave, otherwise the conference will be charged for them, so please hand these in. The Lancet Infectious Disease Commission has been moved to the Alhambra room. And also, don't forget, for those who are members of IUSD, that the General Assembly will be at 1 o'clock in the El Prado room on this floor, El Prado room number 1 on this floor at 1 o'clock. Thank you all, and please join me in giving our 3 speakers a round of applause. We won't have time for questions.